Hello, world. My name's Lindsay, and I'm trying something new today. Okay, so if you've been here, you know that I'm on a research quest. And one of the things that I've been toying with, like the idea of, talking about the papers that I read. Um, so yeah, that's what this is. I'm going to talk about a paper I've read, kind of give my 2 cents on um, how it pertains to research. The way that I'm thinking about it. Um, yeah. And I can already tell that re-going over the paper with the intent of like sharing it with others has dramatically changed how I remember what in the heck is going on in the paper. So I feel like this is a really good thing for me. And hopefully you get something out of it. So yeah. The paper that I want to talk about today is called Drought Stress Memory at the Plant Cycle Level, a review. That's dirt. That's disgusting. That's dirt, though. Okay. Let me see if I can read the names of these people. I struggle with this. Okay. Cécile Haquez, Christophe Salon, Romaine L. Bernard, Vanessa Vernaud, and Marion Prudent. Thank you, guys. This was a good one. And before I get into it, I just want to say that it's amateur hour up in here. I am a Marine and I'm a forester. And both of those careers are really what I like to refer to as like smart dumb. And so a lot of how I process these types of things is smart dumb. And I will often dramatically reduce or simplify the subject, the context of whatever's occurring, so that I can better understand it. So I'm going to be doing that here. So yeah, if I misrepresent any of this information, let me know. I will correct myself, okay? And that would also let me know that I want to talk to you as a human, because if you have any interest in this and you know what I'm talking about, then come on, let's chitty chat. So yes, I don't claim any, I don't lay claim to any of this information. This is other people's work. I'm just talking about it, and I'm talking about it in a smart, dumb way. Okay? So let's do it. Okay, one more thing before we get into it. I just want to say, like, a thank you to these types of people doing these things, okay? Sometimes I read things about climate change and I just get into sort of like an existential crisis mode, right? Because there's not a lot of hopeful things. And don't get me wrong, the people doing that kind of research, the papers that I'm reading, that needs to be done. Light needs to be shed on problems that need to be worked on or fixed. But it can be sad. And then there's times that I read stuff like this and I'm just like, thank God, people are doing things and it's meaningful for us all. And they're just like these little unsung heroes that, I don't know, they're all weird little genetics, ecosystem, plant nerds out working hard, probably obsessively, probably more than they should. But they're doing things that are of benefit for everyone. And so, yes, if you're one of these people, thank you. I appreciate you. So now we can do it. Okay, so this paper is a review of multiple studies. So it encompasses a number of species. Okay, so it was multiple studies that looked at reoccurrent drought and what that did to plant response. Okay. And all of the studies identified something what is known as stress memory. Sometimes it's referred to as like a priming effect. And what they found is that, there was an initial stress, a period of rest, and then another stress, okay? But the plants were changing their response from what happened in the first one to the second one. So there is a sense of like learning, stress memory, and adaptation to stress that caused them to change what they did in the second. Hopefully that makes sense. Okay. So they're basically modifying their behavior from one stress to another. And they found that these stressor events actually enhanced the strength of the plant compared to naive plants who never experienced stress before. So, in essence, it's making them stronger, and they're also learning to alter their behavior to be a better survival response in the second stress. Okay? And you're probably asking yourself, why does this matter? Okay? And it's for this reason. Plants cannot move. They're sessile. They, unlike other creatures, when they experience some form of environmental stress, they can't just get up and move away. We can move them, and that's a tactic that people are exploiting for sure. But the importance of them in being able to regulate their metabolism and their stress response to an environment is key, because they can't escape the environment. They're just there. So that's why this type of research is incredibly important. So yeah, we'll get into the dirty deets. Are you ready? All right, so the study identifies 3 types of stress memory, okay? And in my mind, these 3 types of stress memories can be grouped into 2 categories. So there's the first one, somatic stress memory, and that stays within the plant. The organism that experiences the stress learns to cope in some way, but its offspring will not be able to benefit from that experience, okay? That's somatic stress memory. And then the other 2 types of stress memory, those memories get passed to future generations, okay? And those are called intergenerational stress memory. Now, intergenerational stress memory only gets passed to the first generation. Transgenerational stress memory gets passed beyond the first, so it can go 2 or 3. So those 2 types of stress memory are passed down. Okay? So to loop back to somatic stress memory, this is the one that is within the organism. Okay? And it only lasts the lifespan of the individual organism. This would be sort of like an example of this would be cold hardening. So if you're a gardener, you know that, like, okay, you raise your plants in the house, in your greenhouse, wherever. Before you go to put them outside, you need to acclimate them, in a sense, to where they're going. So cold hardening would be like putting the trees or whatever plant in conditions that are a bit harsher than what they're going to experience. Like you're hardening them. God, okay. So that's somatic stress memory. Now, these types of memories are stored in cells that just make copies of themselves. That's why they're not being passed down. So if you think back to high school biology, cells that just make copies of themselves are through mitosis. They're meiotically inherited. So it's like you have a dandruff flake that falls off your head, your body just makes a copy of that one. Okay, and it's just contained within the organism. This type of stress memory, somatic stress memory, is stored within those types of cells. Okay, and that's how it's not being passed down generationally. Okay, the other 2 stress memories, intergenerational and transgenerational stress memory, those are being stored in cells that are being used in the process of, like, passing genetic information. Okay, so if we think back to high school biology, whatever, meiosis, that's when you have a cell that when it splits, in the end result, it contains half of the genetic material that it needs. It's a haploid cell. Okay, and then what happens is you have 2 haploid cells that come together, and that's all of the genetic material that you need to build the thing. So, like, examples of those types of cells would be like pollen and pine cones. All right, sperm and egg. Those are gametes. Okay, and so that type of stress, the intergenerational and transgenerational stress memory, that's being stored in cells that are like that. Okay, and that's how you're getting that information passed from generation to generation. Okay, and this paper specifically only talks about somatic stress memory. It does talk a little bit about, transgenerational and intergenerational stress memory, but it's kind of just like this is a thing, this is a really basic description of it. Here is the sources that you could really deep dive into that. So if you're interested in that, there's a paragraph on that. Okay. Yeah, and how all of that gets passed down, it's incredibly complex. So I can't even begin to really touch on that in this video. So, okay. There you go. 3 types of stress memory. Somatic, intergenerational, transgenerational. Bada boom. Okay, so the other aspect of this that is highly complex and, what I can't really touch on fully in this video is the process of how these changes are made. Okay. It's called epigenetics. And essentially, how it works is when every organism comes with their computer chip, right? What exists within the genome, okay? That's set from when it very first was created. But what happens is they're finding that certain genes or gene expression can be turned on and off. And that process is so complicated, it's mind-boggling. If you're bored, get on YouTube and look up videos about DNA methylation, histone modification, and chromatin remodeling. Okay? It's fascinating to me that is a thing that is occurring within our bodies, within everything, okay? It's crazy to me that that's happening. It's like weird little organic robots are controlling everything at the microscopic level. It's fascinating. It's fascinating to me that is occurring, but it's equally as fascinating to me that people can figure that out, and then there's people making YouTube videos on it. Like, thank God. So if you're bored, okay, check that stuff out. Maybe I'll get to a point someday when I can explain those types of things in a video, but I'm not there yet. But these are how the modifications are occurring. Essentially, the expression of the genes are changing. Like these weird little robots are opening up pieces of the DNA so that they can be changed. And it's fascinating. So all of this, the changes that I'm talking about are predominantly through epigenetics. So fascinating subject. YouTube it if you're interested. Highly recommend. Okay, so we know that through epigenetics, plants are making these shifts in response to stress. And there's 3 types of that. There's the kind that stays within the plant, and then the 2 that can be passed generationally. Okay, that's what we know so far. Now, what is happening? Like, what are the changes that we're talking about? And so I'm going to try and shed some light on that, okay? So what's happening essentially is that these plants are going through a stress state, and they're creating various forms of metabolites to help them cope with the stress. The metabolites are just a very broad category of things that are regulating the system within the plant to create balance because they're having a lack of that, right? So these metabolites can look like protective proteins, or they can be phytohormones. It's just highly variable. Okay, that's a key thing with this whole process, is this is all highly variable. It has everything to do with the individual species. Okay, so like this paper or this review looked at a number of studies, and so there was like 3 or 4 species, I think, that they talked about primarily. And they talked about how, there was a very small percentage of genes that were similar between all of them. I think they said 18%, but I can't remember which plants. So basically you have 2 plants that are, relatively similar, but only 18% of the genes that they're using to regulate their stress is similar. So it's highly dynamic. It has everything to do with the individual species and the sink-source relationship within that species. So how the plant utilizes energy. Okay, so like, for example, a tuber a tuber that has a potato underground and then the little plant above ground is going to utilize energy differently than a little mustard plant. One of the plants in this is a cute little dainty mustard. It's very petite looking. So how those plants are going to move energy around to deal with the stress is completely different. Okay. So again, one of the best and worst things about science is it's highly variable. There is never a black and white. Okay. So, all right. Lost my train of thought there. It's all highly variable. The metabolites that they synthesize to control things are going to just be completely different from plant to plant, basically. So, right, here we go. Okay, so now I'm going to try and give you like a couple of examples that they gave in the papers of the responses that are occurring, and this is just like a handful of them. There's many more in the paper, and the paper goes into way more depth. Almost too much, okay? And before I get into like what these examples are, it helps me to think about like the equation of plant, right? This is a smart dumb thing that I'm doing here. This is the Marine and the Forester talking. Think very basically about like what makes a plant, okay? This helps me understand the other parts of it. So what makes a plant is nutrients from the soil. You know, the base things that the plant uses to build metabolites. So like iron and boron and stuff like that. Okay, it needs water. Okay, water is essentially what allows photosynthesis to move the nutrients through the plant. Okay, and then yeah, you need light. You need photosynthesis. Okay, that's equation of plant. Now, it's important for me to remember that because I think of it like algebra. Like if you have one piece of the equation that is being altered or changed, the other parts of the equation will compensate to try to equate to balance to what is the best growth, okay? So what's happening is the plants are having their water source cut back dramatically to a point where it's detrimental to the plant. So what does the plant do? It alters the other parts of the equation to compensate. So one example of these metabolites, these things that are being changed is if a plant goes through drought, it will alter its non-photochemical quenching. That's it, you know? You got it? Non-photochemical quenching. So what that means is, this is also a smart dumb thing. I'm absolutely oversimplifying this. This is an entire chemical process that is quite complex. But the non-photochemical quenching is essentially the threshold that the plant decides, right, that once it hits that threshold, it has enough light. It's done. It doesn't need any more. So it alters its state and basically shuts down photosynthetic activity and goes into like a state where it's trying to dissipate heat energy as opposed to drawing energy in. Okay, that's non-photochemical quenching. I have to read it every time because I'll mess it up. And they're finding that the plants, when they go through this initial stressor event, they'll be in their rest period and they can basically alter what that level is so that, right, it hits its photosynthetic level sooner and shuts down, right, to help guard against the loss of water. It's basically shutting down photosynthesis, powering it down so that it can compensate for the lack of water. That's one example of many, okay? And again, these are going to be highly variable and very specific to each species, okay? So another thing that they'll do is try to compensate for that loss of water by creating osmoregulating type chemicals, metabolites, hormones. One of the examples is they'll change the cation composition in their guard cells. So basically, they're closing their stomata so that they're no longer losing water out of the stomata. Okay, that's like a throwback to high school biology. Hopefully, y'all are getting me. Another thing that they'll do is they'll like accumulate solutes, which is like the equivalent of a plant chugging a Gatorade or something. Again, smart dumb. Okay, and they'll create solutes as a means to compensate for the lack of water potential. It's what helps them continue to move water through their body even when there's really not enough to do that. Okay, so they'll do those types of things, but, you know, one of the things that comes from stress events is it's not good for the plants. It's highly toxic for the plants to go through this. So they found that a lot of the genes that are being, you know, activated, changed, whatever, has to do with detoxification of the cells. So they're finding that they'll create protective functions to help them better protect from having damage to the cells. They're enhancing antioxidant enzymes. They're pumping out their own antioxidants. Even plants need them, you know, to reduce oxidative stress. So, and I think one thing that's important, so like, lost my train of thought here. They're creating all of these metabolites, okay? And what's happening is they're going through the stress event, okay, and they've activated these metabolites, and when they go into their rest period, it's almost like the metabolites or the transcripts are in an inactive form. Okay, and then the second stress happens, and the plant is like very quickly able to uptake what's already created in just floating about in an inactive state. So that's another thing that they talk about in the paper. So changes are happening, but they're also accumulating these metabolites in the first stressor event and then using them in the second one. Does that make sense? Hopefully this makes sense. Okay, so I've got a neat little picture here. It used to be nice, but I have children and kids, so it's a good graphic for me. I'm a very visual human. Okay, so the graphic is basically just showing there's an initial stress event, a stress period where nothing's really happening, and then in the second stress event, there's a change. So, you know, this, and it can come in many forms. So it's like, okay, in this initial stress event, there was a down regulation of this metabolite, this transcript, a rest period. In the second stress, the plant recognized, okay, yeah, that was good to downregulate that, but let's downregulate it more. Or it could be the opposite, right? Or there's an event where it's like, okay, we, in the initial stress event, we downregulated this transcript, this metabolite, whatever, rested. And then in the second stress, it was like, okay, this was not good. Let's do the exact opposite of it. Make more, all right, whatever. And then the other scenario where it is a memory gene is where nothing occurred in the first, and then in the second there was a response. And then, of course, you have your null genes that don't make a lick of difference, does nothing. So that's a good graph. You know you're a nerd when you appreciate a good graph. So, yep, we're going to stop there so I can collect myself. Okay, so stress memory is super important for plants to be able to, you know, alter their metabolism in a way that helps them cope with environmental stress, right? But what is equally important is being able to forget that, to turn off that memory. Okay, so like everything, there's trade-offs. So when the plant is expending all of this energy to create protective hormones, phytohormones, or protective proteins and phytohormones, the metabolites, when the plant is dedicating a lot of energy to do that, it is removing energy from being able to just grow normally, being able to yield the food crop or whatever, the cones, okay? So when it is in a heightened stress state, there are definitely negative impacts on growth. So the ability for the plant to just forget that is highly important as well. And there's a whole paragraph in there about it. And I'm sure there are a number of papers that talk about it. So if you're interested in that, check it out. But that is the thing to consider in all of this. And the last thing that the paper talked about is the microbiome interplay. Now, if we go back to the equation of what is plant, the nutrients, the water, and the sunlight, the nutrients all comes from the soil. Okay, and the nutrients that comes from the soil are the basic building blocks that the plant uses to create the metabolites, boron, iron, whatever, okay? All of the things that the plant needs to create stuff, to create the plant. That all comes from the soil. Now, the soil is not just dirt. I don't know if you knew this. The soil is an entire ecosystem. That is the one thing that I got out of my soils class, is that it's not just dirt. Okay, there is an entire ecosystem. You have organisms that are making the base, geology, chemicals available for the plant. There's microorganisms breaking everything down. It's an entire cycle, okay? And it's highly complex. Again, it's dependent on, what the base material is and like what vegetation has existed there previously. It's a whole ass thing, okay? And how the microbiome interplay affects, the ability for the plant to uptake nutrients in stress response, that is all slightly understudied. So if you're interested in that, get on it. So, and, yeah. Science is amazing, okay? And that's pretty much all the paper talked about. They just briefly mentioned seed priming, and this is the first time that I've ever heard of that. The wheat people are doing cool things. They found that if they give seeds a treatment prior to like the stressor event, and like they're finding that it can help them maintain higher growth rate and yield even after the stress. So yeah, the wheat peoples, they're doing good. And that's something that I'm going to follow up on and look at more. I'm very interested in like germination, seed bank type things. So yeah, I'll let you know about that. So that's this paper. Hopefully you found something interesting in here. Hopefully you learned something. I appreciate you for being here this long, if you made it this long. So thank you. And here's the best part, my 2 cents. How it pertains to my research and what I'm thinking about and all that. Everybody's favorite thing, opinions or whatever. So yeah, I think all of this is really cool. It's fascinating. But it is not really possible to do these things in conifers with tree improvement, right? And that's because like these guys that are doing these types of things, okay, they're identifying specific genes and then figuring out what those things are doing. Okay, and they're only able to build the maps and figure those things out because they have these massive pedigrees, meaning that they have like hundreds of generations that they can pull information from, right? Because a lot of like these like small 4B plants or even wheat, I think you can get, at least 1 turn of a cycle in a year, maybe 2. I don't know. I don't know a lot about wheat or rice, but essentially they can get multiple generations in a short period of time. And conifers are not like that. The life cycle of a conifer, right, to go from an organism, like a little tree, to go from a little tree to a sexually mature tree that can make another little tree, that time period is like 15 to 30 years. More often, it's closer to the 30 end of the spectrum than it is the 15. And that's the problem, okay? Places, maybe places in the South that have been working with like loblolly pine, the industrial pine, for years. Maybe they're starting to get at these types of things. But like in the West, We don't have that. We're on like the second or third turn, and it's only for maybe like one or two species, you know. So we don't have the luxury of massive pedigrees to get information from. So that's the first problem. And the genome is infinitely larger. That's a whole, I could talk about that, but probably not relevant for here. It's hard. It's hard with trees, okay? And the thing that makes it hard with trees is the same thing that makes them susceptible to climate change. And it's the fact that it takes 30 years for them to go through a generation, right? There's a paper, a good one, that I can't think of the name of right now. I'm really trying to work on keeping more organized the papers that I read so that I can recall the information or have like the hard copy, you know, as opposed to just my brain. But there's a good paper. I'm going to try my best to find it and link it below. And it's something about trees on the move. I don't know. But the paper looked at like how trees have, like various species have moved across the landscape historically with climatic conditions that existed before. Looking at the time horizon there. And then it shifted and it like looked at more like what is forecasted in the future and will they be able to keep pace. And the question or the answer is like maybe. Again, the beauty of science, there is no like definitive answer. It's more of a spectrum of things, but they think that some species will be okay, you know, the ones that are on the lower end of regeneration time, you know, like 15 years as opposed to 30. Species that are more like geographically spread out are more likely to be okay. So it's highly variable, but the main, people want to debate all of the things and get real into the weeds, but the main thing to take away is that the generation time for trees is long. And it might be so long that they can't keep pace and evolve with the climate. Okay? So what makes it hard to do this stuff makes it, you know, is the danger that they face. So things like bugs will probably be okay because they can have like 3 generations in a year. They evolve much more quickly. They're not sessile. So if you're a bug person, guess what? The bugs are going to dominate. Climate change, probably. Trees, not so much. There might be a cause for concern there. Especially since a lot of bugs mess with trees. Anyways. So, and I do have some ideas of like how to overcome the time horizon. There's breeding without breeding. That's not my idea, but it's absolutely something that we have to do. And I'm going to make an entire separate video on that. It's essentially like building these relationships between like the genomic profile and the trait that you see, the phenotype, and then using computer modeling to model it forward. So in the time that it takes to do one turn, one cycle, you can get 2. And that's critically important. So that's a thing I'm going to be talking about, breeding without breeding. And then I also have some ideas about how we can utilize what is on the landscape. Our forestry practices over the last 50, 60, 70 years, you know, I think there are, you know, things that we can exploit within those practices that will help us get at things in a more timely manner. But I still am formulating those ideas and trying to write them down. So yeah. Ramble, ramble, ramble. I can go on forever. Anyways, I hope you all are well and have a wonderful day, night. Whatever time of day it is. I hope it's good. Yeah.